Hello everyone, welcome to my channel, and today we will be dealing with the DNA barcoding in plants. Okay, let us learn what is DNA barcoding. DNA barcoding is one of the methods for identifying plants to species level. It makes use of a short specific region of the genome like MATK gene or tRNH PSBA like internal transcribing regions, RPOC1, RPOB like DNA dependent RNA polymerase gene subunits, etc. These are all the different types of specific regions of the genomes which are used for barcoding the DNA. And it has got a particular process for barcoding them, and DNA barcoding can be performed first by collecting the materials in need of identification. So it may be any material of our interest. Here if we consider, we have taken a case study of taking a market sample to identify its authenticity to be used as a raw drug or for further processes. So if we use some um, plant material, first we have to collect the plant material which is in need of identification. Then we have to subject it to extraction of DNA from that particular specimen. So that will be the second process in DNA barcoding of plants. And in third process, step of DNA barcoding process is amplifying the sequence, that is internal transcribing spacer region, for all market samples. That means whatever we have taken, the sample what we have collected need to be amplified for a specific region of the DNA. Once it is done and amplified sequencing is done with the amplified sequences, we have to run blast queries for that particular sequence in the online databases. So blast query is online site service which is provided by NCBI, and once we get of blast query results, we will get particular probable results which are comparable for the plant of our choice, and we have to compile a list of probable identifiable candidates for the sample based on the results. So result analysis will be carried out in a fifth stage. So among similar patterns will be getting, but out of this what is the proper plant identify to be identified need to be arrived. To do that, first we have to collect all the identified listed candidates. How to collect? So we can collect fresh samples if we know them, if we have them, if they are available within our surroundings, or even we can take the help of herbariums, or we can take the help of some other means to collect those plants. But we need to collect all the plants in our question, in our list, in our target group, to make it the process further. And again the same process what we have carried stage, we will be extracting total DNA from the reference samples. What are reference samples? The reference samples are the ones what we have listed out and what we have collected for further analysis to confirm the barcoding process. And again we have to amplify and sequence the barcode loci based on our previous set standards. And once we do like that, then we have to create another database for all reference barcodes using a program called formatdb. So again, it is a type of blast and fasta queries only, which are bioinformatical tools which are available in online databases, and using them we have to perform this particular processes. And once we get the blast of each unknown barcode sequence against a reference database, we have to run blast all application so that we will get exact meaning of the blast code. So this is how we can get barcoding. Barcoding is nothing but just like in our day-to-day gadgets, FMCG goods, how we will get the barcodings, that is coded language. This is how, and it should be a unique ID. Just like that, we can obtain a unique identification marker compounds for each and every plants of our interest also. So this is called as DNA barcoding process. Hope you like this particular lecture, and if you like, please like, share and subscribe my channel Plant Science Simplified. Thank you for listening.